Good morning everyone. Today we are going to study about laws of genetics and two crosses, that is monohybrid and dihybrid crosses, and we will correlate these laws with these two crosses. How we can find out laws. The laws were actually not given by Mendel. It was later, when after 16 years of his death, when Correns was studying his work, he formulated these laws. So let us begin today's class. Dominant and recessive. Earlier it was also called as law of dominance, but it is not considered to be a law now. So let us see what was said in that law earlier. There are, let me change the pen. So earlier this was also a law of dominance, but nowadays scientists says that this is not a law. This is what was we were getting as a result. So what happens in this when we consider only one character? Do you remember what do we call that cross where we consider only one character? Yes, it is let us erase that. Now we will discuss what are dominant and recessive traits. When we consider only one character, do you remember what do we call this as? If we consider only one character, what is it called? Yes, monohybrid cross. When we make a cross between pure line dominant homozygous and dominant recessive, what happens in the first generation? This is the parent. Let us consider one of them to be male, one to be female. What gametes will be formed? Gametes, we will have two gametes, but both the gametes will be of same type. Capital T, small t, capital T, small t, capital T, small t, capital T, small t. 4 offsprings are produced in the first generation, that is 4 F1 is there, but all of them are of the same type. For dominant we write capital T, for recessive we write small t. If you remember how we were writing it, T is for tall and small t is for dwarf. But what will happen if we have a heterozygous gene like this? If we have a heterozygous gene like this, only one trait is expressed in F1 generation. Only one trait is expressed in F1 generation. and the trait which is expressed in F1 generation is called, in first filial generation we have only one trait, and trait which is expressed is known as dominant. The trait which is not able to express is known as recessive. Trait not expressed. So that is how we had given the name also to these alleles. One which was able to express, we had taken that word and the first alphabet of that word, uppercase of that, and for the contrasting trait we had taken the lowercase of the same alphabet. So in first generation we get only one of the two traits of this one character, that is two traits are tall and dwarf, we get only tall. So all four are tall. So what will be the ratio, phenotypic ratio? Tall 4:0, where 4 is tall, 0 is dwarf. And genotypic ratio is also same, 4:0, where 4 is heterozygous gene for this. So dominant is the character which trait which is expressed in F1. Recessive is the trait which is not expressed in F1, and on this basis Mendel had selected 7 characters. Let us take one more example over here. Round. When I am writing capital R, that means round is dominant. For this, what will be the wrinkled? Wrinkled will be small r. Wrinkled is recessive. So when we have a heterozygous gene, round will be expressed, right? So this is for when we are considering one character. The dominant which is there, only that is expressed, only one trait is expressed, and that is known as dominant. Wherever we have heterozygous gene, only the dominant will be expressed, the recessive will not be expressed. Now law of segregation. Yesterday we had talked. Okay, so what we will do now, we will do selfing of F, what we received in F1. Selfing. Let us take male, capital T, small t, female, capital T, small t. We will get different types of gametes in this. 2 types of gametes will be produced. So this is G gametes. What are the 2 different types of gametes? Capital T, small t. Capital T, small t. There is a rule for finding out how many gametes can be formed. What is that rule? This states that 2 to the power n, where n is equal to number of heterozygous gene. For example, in this case we have one heterozygous gene, so it will be 2 to the power 1. So how many gametes will be formed? Two different types of gametes will be formed. In this case, capital T, well, how many? We don't have any heterozygous gene, so it is 2 to the power 0. So how many types of gametes are to be formed? Only one. So in the last page we had seen, when we tried to get gametes from this, we had 2 gametes, but both the gametes were of the same type. So one type of gamete is formed when we don't have any heterozygous gene. But if we have heterozygous gene, we calculate according to that. Suppose I take capital R, small r, capital Y, small y. One heterozygous gene, second heterozygous gene. So 2 to the power n. What we will have? 2 to the power 2. So 2 into 2 that is 4 different types of gametes will be formed from this. What are those 4 different types of gamete? Whenever we form a gamete, when we are considering 2 characters, 3 characters, 4 characters, one allele has to be there from each of the gene. So it is capital R, capital Y, capital R, small y, small r, capital Y, small r and small y. We will discuss this in more details when we will study mono-dihybrid cross. So this is the formula for finding out how many different types of gametes will be formed when we are doing any cross. So gametes, this is two different types of gametes had to be formed, and we have 2 different types of gametes. Now what will be the F2? F2 says small t. So what does law of segregation states? Law of segregation states that during gamete formation, all the alleles segregate out. We will have only one allele in one of the gamete. So all the alleles segregate out during gamete formation. Now what happens because of this segregation? How can we show that they have segregated out? Now in F2 generation we get phenotypic ratio phenotype. Do you remember? The one which we can see from our eyes. These 3 are tall. So tall is to dwarf is 3:1. This is phenotypic ratio. But if we consider genotypic ratio, it is capital T is to capital T small t is to small t, and we have only one capital T over here, so it is 1 is to, we have 2 capital T small t over here, 2 is to, we have again 1 small t. So genotypic ratio is 1:2:1. Now we had only tall in the F1 generation. How could we get this in the F2 generation? We got this because during gamete formation, alleles had separated out. So this is law of segregation. This is how we can say law of these gametes have segregated out, and law of segregation states that during gamete formation, the alleles of a gene separate out. Next is Punnett square. Punnett square is expression of a cross in the form of a square. On one side, we write all the male gametes, on the other side we write all the female gametes. So for this cross, let us write it for capital T into capital T small t. What is the gamete formed? 2 different types of gametes are formed, capital T small t, capital T small t. So what is for male? It is capital T small t. For female, it is again capital T small t. How do we write it? We cross these two, capital T, capital T. As we write in mathematics T square, we don't write like that in biology. We will write both of them together, capital T and small t, capital T and small t, small t and small t. So it is representation of the same cross in the form of a square, and this is known as Punnett square. So what we do, on one side we write the male gametes, on other side we write the female gametes, and then we cross it with each one of it. Okay, this one with this one will be written over here, this with this will be written over here, this with this will be written over here, this with this will be written in the second line, this with this will be written over here. So this is how we represent a Punnett square. Law of independent assortment. This law can be proved by a dihybrid cross. Dihybrid cross. Now, what do we mean by dihybrid cross? That means we are considering two different characters. Two different characters we are taking. So let us take tall and yellow, dwarf and green. Yellow seed color is dominant and tall stem height is dominant. So because we don't have any heterozygous gene over here, how many types of gamete will be formed? Only one type of gamete will be formed, so I am writing it only once. Here also no heterozygous gene, so only one type of gamete will be formed, that is small t, small y. When we get F1 generation, all of the plants will be of same type because same type of gamete is formed, capital T, small t, capital Y, small y. If we have capital T, small t, capital will be expressed, so all the plants will be tall and all the plants will be yellow. They will have yellow seeds. Tall plant with yellow seeds. This is what we get in the F1 generation. Only the dominant character is being expressed. Now on selfing, what will happen? Capital T, small t, capital Y, small y into capital T, small t, capital Y, small y. How many types of gametes will be produced? We have two heterozygous genes, so gametes that will be produced is 2 into 2, that is 4. Let us see how. So we have capital T, small t, capital Y, small y into capital T, small t, capital Y, small y. 4 types of gametes are formed. I told you one from this character, one allele from this character, and the other allele should be from this character. So we have capital T, capital Y, capital T, small y, capital T, small t, capital Y, small t, small y. So these are 4 different types of gametes that are formed when we have this as parent. Here also we have the same type, so we will have same 4 gametes, capital T, small capital Y, capital T, small y, small t, capital Y, and small t, small y. So what will be the F2 generation? Let us draw a Punnett square. Again, I am not drawing it well. I am not used to these graphic tablets. All the line that I am drawing is coming out as if Okay, so this is my imperfect square. Anyways, when you draw it perfectly. So this is the male gametes we will write on this side, the female gametes we will write on this side. So it is capital T, capital Y, capital T, small y, small t, capital Y, small t, small y. Capital T, capital Y, capital T, small y, small t, capital Y, small t, small y. Now how do we write? I taught you in the monohybrid cross, we will write, see these two and then write capital T, capital Y, then see these two, then write capital T, capital Y, small y. See these two, capital T, small t, capital Y, and then see these two, capital T, small y, capital Y, small y. You will see that all these four will have same. Let me write first, then I will tell you. Capital T, small y, capital T, small t, capital Y, small y, capital T, small t, small y, small y, capital T, small t, capital Y, capital T, small t, capital Y, small y, small t, capital Y, small t, capital Y, small y, capital T, small t, capital Y, small y, capital T, small y, small y, small t, capital Y, small y, small t, small y, small y. Now let me change the color of the pen. You see, an easy way to write it. This, as we will proceed from this to this, this to this, this to this, all of them have the same character. And capital T and capital Y, if they are having capital T and capital Y in any of them, that means the dominant characters are to be expressed. So 4, 5, 6, 7, 8 and 9. So in 9 of them we had same characters that were expressed. That is tall, and yellow seeds. How many plants are there? 9. Now if we see how many plants are there, we started with tall and yellow seed. How many plants are there which are dwarf, and wrinkle seed? Dwarf and how many? Only one. Now what about the remaining? There are many of them which are still there. So what about the remaining ones? See, we have capital T in this, no capital Y. Capital T in this also, but no capital Y. And capital T in this also, but no capital Y. So this means This means tall is there but with wrinkled seeds. How many are there? 3. Tall with wrinkled seeds, 1, 2 and 3. Let us see the remaining 3, what is their characteristics. 1, no capital T is there, no capital T, no capital T, that means they are dwarf. So dwarf, but they have capital Y, and yellow seeds. How many? That is also 3. So what is, we started with tall and yellow seeds and dwarf and wrinkled seeds, but we got 2 new characters when we saw the F1 generation. What does it say? When even if we are considering 2 characters, so what is the ratio? Tall and yellow 9 is to tall and green 3 is to dwarf and yellow 3 is to dwarf and wrinkled 1. So this is the parental character, this is also parental character. These are the characters with which we started. So these are parental characters. What are those 2? Tall and yellow and dwarf and wrinkled. But we got 2 new characters and they are called recombinant characters. What are those? We got 2 new characters and these are recombinant characters. What are those? These 2 are new characters that we got. So tall and green and dwarf and yellow. These are recombinant characters that we got. From where did we get this recombinant character? This is what law of independent assortment is all about. Law of independent assortment says that when we are considering 2-3 characters at a time, during gamete formation, they are independently assorted out. It is not that they will be linked together. They are independently assorted out, and this is what is law of independent assortment states. So that is why, and how is it proved? We get 2 new characters. Had it not been independently assorted, we would always get the same parental characters. But as we are getting 2 new characters, this suggests that during gamete formation, the characters are independently assorted out. So hope you got it. So this is all for today. We will come again with something new tomorrow. We'll proceed with the chapter tomorrow. Thank you for watching. Enjoy learning. Subscribe it and like it. Share it with your friends if you liked it.